welcome to today's session. This is for the session disclaimer, and I personally have no relationships to disclose. Welcome to the session, Metabolic Crosstalk between Pathogens and Hosts in Cystic Fibrosis. Me, Hazel Osuna, and Sebastián Riquelme, we're going to be sharing this session, and I'm excited to talk about the talks today. We're going to be looking at the different impacts. For example, you have the pathogens, different pathogens in the lung environment, and however, we also know what happens on the other side, what the host or the immune cells do. However, we still know very little about the signals or this crosstalk between host and pathogen. And these are the points that we're going to highlight today in today's session, discussing novel mechanisms of bacterial survival in the CF airway. Also, how pathogens exploit these metabolic platforms, orchestrating inflammatory response to infection, or also coined as immunometabolism. We're going to also examine how these metabolic circuits are altered in CF phagocytes and how these changes increase the susceptibility of people with CF to develop chronic bacterial colonization. So I'm going to present our speakers for today. We have Sahil with immunometabolic control of innate memory during Staphylococcus aureus and Pseudomonas co-infections. We also have Jen with immunometabolic regulation of epithelial macrophage crosstalk in CF, and Sebastián with mitochondrial regulation of tolerance to bacterial lung infections. And our last talk for the session is going to be Neha with metabolic approaches to probe interaction among microbe host drugs in genus Burkholderia. Now, I'm going to present our first speaker. It's going to be Dr. Sahil Mahajan. He's a senior researcher, scientist at Ohio State University, and he works with Dr. Daniel Wozniak, and they work with Pseudomonas and co-infections in CF and also metabolism.
How one bacterial pathogen makes it easier for the second bacterial pathogen to infect the host, and how this phenomena is controlled by metabolic reprogramming of infected cells. I have nothing to disclose related to this presentation. So now this graph highlights global estimates of mortality associated with 33 clinically relevant bacterial pathogens across 11 different infectious syndromes. These pathogens are altogether responsible for 15% of all global death and 55% of all infection-related death for the year 2019. Two pathogens that are relevant for today's talk, Staphylococcus aureus and Pseudomonas aeruginosa, are ranked first and fifth in this mortality estimates linked to bacterial infection. And together, these two pathogens are responsible for roughly for the loss of 2 million deaths, 2 million lives every year. So clearly, Staph and Pseudomonas have deadly impact on human health. They infect both males and females, and they infect people of all age group, right from infants to the elderly. So irrespective of the gender or irrespective of the age, anyone can be infected with Staph or Pseudomonas. And because these two pathogens infect such a broad range of individuals, the mortality associated with them is extremely high. Another notable characteristic of Staph and Pseudomonas is their ability to coexist. In fact, one of the most prevalent polybacterial infections studied in the lungs of CF patient is with Staph and Pseudomonas. And this point is highlighted in this graph where you see that Staph and Pseudomonas are prevalent in initial layers of CF, and their number increases over time, which in this cohort goes from 30 to 50%. And this was the era before ETI was introduced. And now what happens with ETI when people with CF starts on ETI therapy? So we see some benefits in initial few months of the ETI therapy, but minimal further improvement in the later stages of the infection. So suggesting even for those patients that are on ETI therapy, the presence of staph and Pseudomonas poses a significant challenge. Now, there is a lot of data that has emerged in recent months that highlight this point that polybacterial infections result in poorer outcome compared to monobacterial infections. For example, this graph on the left suggests that if the patient that suffers from polymicrobial Staphylococcus aureus bacteremia has worse 6-month survival compared to patients that suffer from monomicrobial Staphylococcus aureus bacteremia. Again, the graph on the right suggests that patients with polymicrobial Staphylococcus bloodstream infection has worse in-hospital mortality compared to patients with monomicrobial Staphylococcus aureus bacteremia. And interestingly, in both these studies, among polymicrobial staph patients, in 90% of the cases, the second pathogen that is detected is another bacterial species. So this really led us to ask this question: does polybacterial infection influence the way our immune system responds? And macrophages contribute to host defense against both staph and Pseudomonas. And in fact, macrophages are the first immune cells these bugs sees when they first breach the host defense. These macrophages are anatomically located everywhere in the body where they perform very specialized functions. For example, the alveolar macrophages, which are the macrophages that are present in the lungs, have a great role to play in maintaining the integrity as well as fighting respiratory infections. And if you look at the role of alveolar macrophages in CF patients, what has been shown that in the lungs of the young children with CF, the number of these alveolar macrophages goes up even without any infection. And this number further increases upon infection, suggesting that macrophages had an important role to play in fighting these respiratory infections. So we wanted to really investigate the role of macrophages in polybacterial infections. And for that, we took this bone marrow-derived macrophages and infected them with USA 300, which is the reference strain of staph we used in our experiment. We infect these macrophages for 30 minutes. We kill any extracellular bacteria with gentamicin. And after an hour, harvest these macrophages to see what is the percentage of cells that get infected with the bacteria. And what we see is after an hour, roughly 85% of macrophages have bacteria inside them. And then with every passing day, we see this overall percentage of infected cells goes down. Then we looked at what happens to the intracellular bug burden. And again, we see that you have maximum burdens during first few hours after the infection. And then as the disease progresses, the intracellular bug comes down. And by somewhere on day 4, these macrophages have completely cleared the infection. So what this suggested is macrophages are effective in phagocytosing the bacteria, and they are also very effective in killing it intracellularly. So then we looked at what happens to the activation of macrophages in response to staph infection. And two of the pro-inflammatory markers I'm highlighting here is TNF-alpha and IL-1 beta. Their expression goes up 6 hours after the infection, but interestingly, by 24 hours, the expression has already come back to the basal level. We also look at some anti-inflammatory markers, and one of them I'm showing here is, thank you. And one of them I'm showing here is aeruginosa 1. So the expression of this anti-inflammatory marker lags behind the pro-inflammatory marker. It comes a little late. Its expression gets induced, say, by 24 hours. But interestingly, even the expression of ARG1 has fallen down by 48 hours. So if you look at the overall magnitude of innate immune response, what we see that these macrophages are very rapid in responding to the infection. The response peaks at around 12 to 24 hours, and then it gradually comes down. And by 48 hours, it has already come back to the basal level. So this is the time we decided to hit them with second bacteria. And we really asked this question that can primary Staph aureus infection alter macrophage response to secondary Staph aureus infection? And this is the experimental design we followed. We have 4 groups. We have macrophages that never see any infection. Then we have macrophages that get only first infection on day 0. Then we have third group where macrophages get only the second infection on day 2, and the fourth group that got first infection and then gets reinfected 2 days later with the second round of infection. And this is what we see. So control group here does not produce any pro-inflammatory markers, and 3 of them I'm highlighting here, TNF-alpha, IL-6, and IL-1 beta. The macrophages that got only the first infection by 60 hours, they have already come back to the basal level, and again, in second group, we don't see anything. The macrophages that got only the second infection, we see induction of these pro-inflammatory cytokines in their supernatant, so all of them are elevated. But interestingly, the macrophages that got first infection with USA 300, when we reinfect them again with USA 300, they completely lose their capacity to produce these pro-inflammatory cytokines, TNF-alpha and IL-6. The levels of IL-1 beta is significantly reduced. Then we wanted to look at what happens to their phagocytic potential, and again, the macrophages that see the bacteria for the first time, roughly 85% of them get infected. But compared to that, the macrophages that have gotten the first infection with USA 300, when we reinfect with the second round of infection, their capacity to phagocytose has significantly reduced. And then we looked at a lot of other pro-inflammatory markers, and again, I'm highlighting a few of them here. We have pro-inflammatory cytokines. We also have certain chemokines, and one of them I'm showing here is CXCL1, and this gene NOS2 that encodes for an antibacterial gene, inducible nitric oxide. So again, we see the macrophages that see the bacteria for the first time, they are very effective in inducing the expression of these pro-inflammatory markers. But first infection really takes away their capacity to fight the infection, to fight the second round of infection. So then we wanted to look at all right, their pro-inflammatory markers are down, so at the same time, are they inducing anti-inflammatory programs? And then we looked at a lot of anti-inflammatory genes, and again, 5 of them I'm highlighting here. So it looks like their pro-inflammatory markers are down, but they haven't induced their anti-inflammatory programs. So then we wanted to ask this question, do we see this response if we challenge this with totally unrelated bacteria for the second time? And we chose Pseudomonas for that because, one, that it engages totally different receptor on the surfaces of macrophages, where Staph mainly signals through TLR2. Pseudomonas goes through TLR5 because of their flagella or LPS that engages TLR4 on the surface of macrophages. And then, again, we had 4 groups. We have macrophages that never sees the infection, macrophages that get first infection with Staph, macrophages that got only second infection with Pseudomonas, and the 4th group of macrophages which get first infection with Staph and get second infection with the Pseudomonas. And again, we find the same thing, macrophages that are seeing Pseudomonas, they really induces these pro-inflammatory cytokines, TNF-alpha and IL-6. But those macrophages that have seen the first infection with USA 300, when we re-challenge them with Pseudomonas, their capacity to produce these pro-inflammatory cytokines is severely reduced. Again, we looked at the phagocytic potential, and we found that macrophages are now very effective in taking up bug. Only 6 to 7% get infected. But those macrophages that have first received this USA 300 infection, when we challenge them with POA1, which is the reference strain of Pseudomonas, we see 50% reduction in the phagocytic potential. And again, the same story, the macrophages that are seeing the Pseudomonas for the very first time, they really upregulate these pro-inflammatory programs. But those macrophages that have seen this previous infection with US 300 really lag behind in inducing this. And again, anti-inflammatory programs does not go up in them. So then, if I conclude my first round of what I have talked here is, so what we have shown is that primary infection with Staph aureus induces a tolerance phenotype. So this suppressive capacity of macrophages in immunological terms, we call that as tolerance. And it does not matter what is your second infection, whether it's a homologous bacteria or a heterologous bacteria. And then we did a lot of experiments. We thought maybe these macrophages need more rest in between 2 infections and they will recover. So we really went between two infections. We increased the resting period from 2 days to 4 days. And again, we saw that tolerance phenotype is maintained. We then switched the bacteria. We gave first infection with Pseudomonas and then re-challenged with Staph. And again, we saw the tolerance phenotype is maintained. And then we got macrophages from other anatomical location just to rule it out that this is not something very specific with VMDMs. And again, the tolerance is maintained. We also made bone marrow-derived macrophages from healthy donors. And even in those macrophages, we saw the tolerance phenotype. So far, what our model shows that if you have naive macrophages, they're gonna, and if they see the bacteria, they get activated, they try to clear the infection. And during this process, they get into this altered state where they have the memory from their first encounter with the bacteria. That then takes away their capacity to fight the second infection. So we wanted to understand what is different about this altered state that led them into this tolerance phenotype. And for that, we did 8x-seq on these macrophages. So what 8x does is it provides us a genome-wide map of accessible chromatins. So when we look at the whole accessible chromatin regions of promoter region in all the genes coming either from naive bacteria or these macrophages that have been infected with USA 300 and has induced a tolerogenic phenotype. We saw subtle differences, and when we looked for the pathways on these enriched regions, we saw a lot of different pathways get enriched for these chromatin regions. But two of the pathways that I'm going to talk today is one is the metabolic pathway, where we saw maximum number of genes get changed. And the second is this pathway of neurodegeneration, which I'll talk a little bit in the last half of my talk. So because the metabolic pathways were different in this altered state, so we then went ahead and did the metabolomics on these 4 groups of macrophages. And when we did PC analysis on them, we found that all the 4 groups have different metabolic profiles. So we have these uninfected macrophages here. Macrophages that got only the first infection of USA 300 is here. Macrophages that got only the second infection with POA here is, and the macrophages that got both the infection is somewhere in between. And then when we looked at the pathway, we saw a lot of sugar-related pathways, the nucleotide pathway, as well as the amino acid pathways are changed. And then when we looked at the top 5 enriched metabolites in this tolerant macrophages, we saw a lot of metabolites related to TCA cycle are enriched. There was oxaloacetate, malate, fumarate. So then if you look at the TCA cycle, so we get all these metabolites, but the end product of this TCA cycle is this NADH molecules and FADH2. So this NADH then transfers its electron to the complex 1, which is NADH dehydrogenase. FADH2 transfer its electrons to complex 2, and then through this electron transport chain, they really pump this hydrogen outside and generates a mitochondrial membrane potential. So if you have a increase in the TCA cycle, it normally increases the mitochondrial membrane potential of the cells. So we then went back and looked at the mitochondrial membrane potential in these macrophages. And again, these macrophages that have this tolerogenic phenotype induced, they have increased fluorescence with this dye that tracks the mitochondrial membrane potential. And then finally, we really wanted to talk, is this change really relevant to our model? So for that, we blocked this OXPHOS pathway of ETC chain by using either rotenant that blocks the complex 1 or oligomycin that blocks the TERT5. And what we see that, again, we see when we give PO1 infection, we have induction of this cytokines IL-6 and NOS2. So macrophages that have seen a previous infection of USA 300 when get re-challenged with PO1, they have gone into tolerogenic phenotype. And then those macrophages in between 2 infections, if we treat them with either retinone or oligomycin, we see that their expression comes back. So we really reverse the tolerance if we block OXPHOS. And then we really looked at the overall health of this mitochondria. And what we were very surprised to see that macrophages that are undergoing this tolerogenic phenotype has this long chain of mitochondria in them. It seems to us that many mitochondria have come together to form these long chains. And then we can really, in the cells that this mitochondria can do form these big chains by undergoing this program called mitochondrial fusion and fission, where you have these two adapters, mitofusions on the surface of the mitochondria that really let them come closer and allows them to fuse. Or sometimes if the mitochondria is not in good health, they can also undergo this fission program with certain adapters like the DRP1 and PINK1. And apparently, when we went back to our ATEX-seq data, we found that this pathway of neurodegeneration has really enriched for all those genes that are involved in the mitofusion program of the mitochondria. So I think at this stage, we are really looking into the role of this mitochondrial dynamics with fusion and fusion, its impact on OXPHOS and its impact on the tolerance phenotype. So overall, what I have shown today is that if these macrophages get a primary infection of Staph aureus or Pseudomonas, they induce this tolerogenic phenotype. And it does not matter what your secondary infection is going to be, whether you infect them with a homologous bacteria or a heterologous bacteria. We also showed that the induction of tolerance in infected macrophages is associated with cellular changes in cellular metabolism. And so far, we have shown that if you block OXPHOS, you reverse this tolerance in infected macrophages. And with this, I'm really going to thank everybody from the worsened group that has really been helpful in getting this project started. And all the metabolomics data that I've shown today has come with the help of Haitop Wen from OSU and John Asra from Harvard. And we have a great C3 core and whose support has really been helpful in getting this project going. And with that, I'll be happy to take any questions.
Any questions from the audience? Unfortunately, we don't have a microphone, so if you have a question, please speak out loud.
So we haven't really looked at the expression of phagocytic markers, or for that matter, DLRs that probably be engaging on the surface of these macrophages. And that's one possibility that their expression is downregulated and they're not able to recognize the second round of infection. So that's quite a possibility. We haven't really looked into that. And answering your second question, so we also looked at different size beads. And again, we see the same effect there. So their phagocytic potential is compromised. So it's not only with bacteria, but if you take anything which is not live, even there also we see this phagocytic potential is compromised. I think what you are saying is partially right. It depends at what stage of infection we capture these macrophages. So if you take on this, if you take and do the metabolomics very early on in the infection, you see a lot of glycolysis is up. We see a lot of lactate because pyruvate is not moving into the OXPHOS. But gradually, I think the glycolysis comes down and the macrophages make a shift toward OXPHOS. That's quite a possibility that if we reverse and make them get more into glycolytic pathway, we might not see this tolerant phenotype.
Another question there on the back.
So answering your first question, we have done all those experiments where we did heat-kill bacteria. We also purified lipopeptides from Staph aureus cell wall and did experiments only with them. So we see even with those tolerances maintained, even though the extent of tolerance is very different when we have live bacteria versus where we have heat-kill bacteria. Answering your second question, that if we directly induce OXPHOS in these macrophages, can we then get away with first round of infection? So that's the work we are doing there, that we are trying to find out if we can pool certain metabolites, if we then give it to macrophages and induce tolerance phenotype, irrespective of whether the infection is there or not. But I don't have a real answer for that at this stage.
A question from the audience? Okay, so I think we still have time for questions. So I have another question for you. So you mentioned that, first of all, great presentation. Thanks very much. You mentioned the role of the preservation of OXPHOS to maintain this tolerogenic environment. And you describe everything in macrophage, but OXPHOS is also important in every single cell in the body.
Yeah.
So, and every single cell in the body is responding to the diet of what we are eating to produce energy. So do you have an idea how the diet of the host is influencing this tolerogenic phenotype to the infection?
So one of the questions, every time I went and took this data to Dan, he has this question, like if what we are seeing is really true, that why we don't see it in humans, because they get exposed to this pathogens all the time. So what it is that how they reverse the tolerance, and I think the answer lies in the diet. But at this stage, we don't have. We have certain hits from our ATEX-seq data, and now we are in the process of getting chip sector to really understand. So the question was, can we do some sort of dietary interventions and reverse this tolerance phenotype? And right now, we don't have an answer for that, but it's quite possible. And now we have this ATEX-seq data. We are also in the process of getting chip-seq done to really understand all the metabolic enzymes that might be playing a role and try to figure out which pathway might be the most relevant and how to play around with that. Okay.
I think
Thank you.
Okay, thanks very much, Said. Yeah. So it's my honor to introduce the following speaker, Dr. Jennifer Bomberger, Professor of Microbiology and Immunology at Dartmouth Geisel School of Medicine. In her laboratory, she works with Pseudomonas staph, different pathogens, inflammation, and metabolism. So please, Jennifer.
Okay, thank you so much. I appreciate the opportunity to present, and thanks to Hazel and Sebastian for inviting me. I'm going to talk today about some work we've been doing looking at epithelial macrophage crosstalk in the respiratory tract, particularly in the setting of viral bacterial co-infection. I have no disclosures. So we all know in cystic fibrosis, we've got very frequent infections in individuals with both Staph aureus and Pseudomonas aeruginosa. In our laboratory, we're really interested in understanding how these chronic infections get established because they're so important to the outcome, long-term outcome of people with CF. And so my laboratory has been very interested in the contribution of the mucosal environment and how the respiratory epithelium and the host is really important in mediating these conversions in people with CF to chronic bacterial infections. And so when I started my lab, we became interested in clinical data suggesting or showing correlation with acute respiratory viral infections when patients develop chronic bacterial infections. And so I'm showing data here with studies looking at the acquisition of a chronic Pseudomonas infection. And if you look at the months of the year, you can see that the months where patients get predominantly chronically infected with Pseudomonas is during the winter months. And so we all know that is respiratory virus season. We know in individuals with cystic fibrosis, acute respiratory viral infections account for about half of their pulmonary exacerbations. Individuals with CF get about the same number of viral infections as you and I every year. They tend to be worse with previously hospitalizations and worse respiratory symptoms. These infections tend to be most commonly are influenza, rhinovirus, and respiratory syncytial virus. It's been shown in a number of different clinical studies now. I'm just showing a few favorites here that if you look at Pseudomonas colonization, about 85% in this study of Pseudomonas colonization happened within 3 weeks of an acute respiratory viral infection. And in this study, we showed RSV infection, or it was shown, we didn't show. RSV infection was specifically associated with the acquisition and the development of chronic Pseudomonas infections. And so we've become very interested in my lab in trying to understand how acute respiratory viral infections impact the development of chronic bacterial infections. And so I'm going to show you data here with Pseudomonas aeruginosa, but we've seen very similar things with Staph aureus as well, where we grow polarized human airway epithelial cells. And if we perfuse these airway epithelial cells with media, we can inoculate them with Pseudomonas. Like I said, same thing with Staph aureus, and let aggregates form in what would be the lumen above the airway epithelial cells, similar to what happens in vivo. And we see with mock infections, we get aggregates of bacteria. This is a top-down view now. Aggregates of bacteria above the respiratory epithelial cells. But if we give a preceding viral infection, similar to what's seen in patients, we see dramatically enhanced Pseudomonas biofoam growth. So my lab has been really interested in trying to understand the mechanism of how these co-infections can promote chronic infection. And we've summarized a whole lot of work here in a couple sentences. We've shown that dysregulations in nutritional immunity, and particularly iron bioavailability, are really important, are one factor that's important in promoting secondary bacterial growth in Pseudomonas biofilm in response to an acute respiratory viral infection. And then we've recently shown, looking at the interferon response, so looking at the antiviral response alone in the absence of the virus, just antiviral signaling can reprogram the metabolic state of the respiratory epithelium, causing an increase in glycolysis and lactate release, which can also promote Pseudomonas biofilm. So we know some of these metabolites and nutrients are more prevalent in the setting of co-infection, and that appears to promote Pseudomonas biofilm. We've also gone on to show that one mechanism cell biologically of how these nutrients are getting released is in association with extracellular vesicles released from the respiratory epithelial cells. These are released in the presence of both a viral infection or just induction of antiviral signaling. And in a previous publication, we've shown that these vesicles intimately interact with Pseudomonas aeruginosa. I'm showing you images here with fluorescently labeled Pseudomonas aggregates. You can see here, if we fluorescently label extracellular vesicles, we see they co-occur where the Pseudomonas biofilms are. So these bacteria in the human vesicles are being intimately interacting, and we've shown in this previous study that they promote biofilm. So what are extracellular vesicles? So these are vesicles that are released from most cell types that have actually been studied. They come in different flavors, small EVs, large EVs. This is dependent on how they're actually being released from the origin cell. They then transport cargo. This is a variety of different cargo. It can be small RNAs, lipids, proteins, all metabolites, all kinds of molecules can be released in these and transported to recipient cells where they can influence the physiological function of that recipient cell. We know the cargo has been shown to contain infectious material. It's also been shown to contain, though, and transport important antiviral immunity molecules as well as antibacterial immunity molecules. And so when we started this study, we were interested in its role in co-infection. And so these extracellular vesicles have not been studied in co-infection. And so when Sydney Lane joined the lab, she's an immunology student, she was interested, as we've shown, that these vesicles interact with Pseudomonas. Sid was interested if these vesicles are actually interacting with innate immune cells that might be in the area, knowing that this is another cell type that would be prevalent in the airways in the setting of co-infection. And Sid was very interested in the role of macrophages. We know these are a very abundant cell type in the respiratory tract. We know they're really important in controlling bacterial infections. And it's been previously shown that epithelial extracellular vesicles have been shown to preferentially interact with macrophages. And so that drew our interest in looking at these vesicles being released from epithelial cells infected with virus or undergoing antiviral signaling and seeing how they may impact co-infection. The other thing that drew our attention is it's really well known in the co-infection literature, less CF co-infection, but general co-infection in healthy individuals, that macrophages are highly dysregulated. And this is an important part of the dysfunction that happens during co-infection that permits the secondary infections to develop. And so we know that there's aberrant inflammatory signaling during co-infection in macrophages, and they have an impaired ability to clear bacterial pathogens. So for these reasons, we wanted to study macrophage interactions with these vesicles. So Sid's hypothesis when we started this study was, do extracellular vesicles released from the epithelium during the antiviral immune response contribute to macrophage dysfunction during co-infection? And so we decided to focus on the antiviral immune response to remove the potential that there were viral proteins being secreted in these vesicles. And so the data I'm going to show you today is using a model where we've treated airway epithelial cells with poly-IC. This is a viral mimetic. It's double-stranded RNA. Depending on how you treat your cells, whether you float it or transfect, you can engage different pattern recognition receptors, which will initiate your antiviral immune program in the respiratory epithelium. So the first thing we did was we treated our airway epithelial cells, polarized at air-liquid interface with poly-IC, and asked, do we see an antiviral signal in the epithelium? And we do. And so I'm showing you just a suite here of antiviral cytokines that we would expect to change. We see strong induction here of type 1 and type 3 interferons, which are your classic antiviral signaling molecules. So now that we knew we had an antiviral program being induced, we asked, control extracellular vesicles versus antiviral extracellular vesicles, do these have any impact on macrophage function during co-infection? So this is our schematic of how we prepare our extracellular vesicles. We incubate poly-IC with the respiratory epithelial cells and over 18 hours collect the apical secretions, meaning any vesicles that have been secreted over this time period. We then use differential centrifugation to purify both large extracellular vesicles and small extracellular vesicles. And we combine those for the studies I'm showing you, trying to think about both vesicles would be present in the airways during a co-infection. And so that's what we wanted to model. So the first thing we need to do is kind of characterize our extracellular vesicle population, both in control extracellular vesicles. So just the lingo here, control extracellular vesicles or vesicles released from control epithelial cells, antiviral EVs or extracellular vesicles. I'm going to use this term, meaning those are the ones released from the epithelial cells. This is going to get a little complicated when we start adding in macrophages. So I just want the terminology to be clear. So when we looked at protein content, mean extracellular vesicle size or particles, number of particles, we see no significant differences in control versus antiviral extracellular vesicles. And so this was important to us to suggest if we see macrophage phenotypes, that suggests it's something different in the cargo, not just different numbers of vesicles being released. So our first question was, do these extracellular vesicles interact with macrophages, and is that different during during an antiviral program? So the way we decided to do this was using flow cytometry and imaging. I'm going to show you flow cytometry data. We labeled extracellular vesicles by using a cell trace far red, so a cell marker that would be taken up by the epithelial cells, and then as they produce extracellular vesicles, those vesicles will now be fluorescently labeled. We can then collect, as I showed you, collect the extracellular vesicles, purify them, and then apply them to macrophages. We're using primary human monocyte-derived macrophages for our studies, in part due to the work by Clemente Brito showing that recruited macrophages are an important cell type in the CF airways. So that's our cell type. So if we incubate these control and antiviral extracellular vesicles with macrophages, we see, interestingly, I'm showing the flow plots here, but quantified, we see, interestingly, no difference in the association or the uptake of these vesicles in the macrophages. And so we do see association, but it's not different in control versus antiviral extracellular vesicles. So the next question was, do these antiviral extracellular vesicles actually impair or alter macrophage phenotype in any way? And so the way we decided to do this was to use an antibiotic protection assay. And so we took our extracellular vesicles, incubated them with macrophages. After an 18-hour period, we then exposed them to bacteria. I'm going to show you a couple different bacteria we used, and then we performed an antibiotic protection assay. We can look at uptake and survival in the macrophages. And I'm going to show you data with CFU remaining in the macrophages, which is a ratio for us of the bacteria that survive in the macrophages as a ratio of what was originally taken up. So killing capacity of the macrophages. And when we do that, if we look at USA 100 is a strain of Staph aureus and PA01 is a strain of Pseudomonas aeruginosa, we see an increase in CFU remaining of Staph aureus when the macrophages are exposed to antiviral extracellular vesicles, suggesting impaired clearance of Staph aureus by these macrophages. We also see a similar trend for Pseudomonas, but this trend never reached significance in our studies. So the work I'm going to show you moving forward is going to be with Staph aureus. We've also repeated the same findings with, this is a USA 100, a hospital acquired. We also used USA 300 strain, and we see very similar phenotypes. And these responses, this impaired macrophage killing, is not just due to overall changes in viability of the macrophages. They're all surviving to similar levels. So now we wanted to understand what is the mechanism that's underlying this impaired killing. And so we looked at macrophage polarization. I know this can be a polarizing topic, pun intended. Macrophages, we know, are highly plastic. They respond to environmental cues. This is a spectrum. This is a way of thinking of them functionally. This is not M1 versus M2 strict phenotypes. But this is helpful when we're trying to think about macrophage function and its ability to clear pathogens. And so ways we can think about the functional state and characterizing changes in the functional state of macrophages to look at cell surface markers or cytokine secretion. And so that's what we did with macrophages that have been treated with control or antiviral extracellular vesicles. So I'm showing you here our assays looking at inflammatory markers, so cell surface markers for inflammatory macrophages. And kind of surprisingly to us, we saw no difference in different cell surface markers for the inflammatory state for macrophages with control versus antiviral extracellular vesicles. We did these experiments in the presence and absence of Staph aureus challenge to see if that had a phenotype, and it didn't. And we also looked at pro-resolving markers. In the interest of time, I'm not showing them, but those also weren't significantly different. And so then we decided to take a look at cytokines. So maybe the cytokine secretion is different in these cells. And we were somewhat surprised to see if we compare control to antiviral extracellular vesicle treated macrophages, we saw very little change in our macrophage secretion profiles, slight change in RANTES, and a more significant change in IL-6. And these are the actual cytokine concentration changes that we saw, but very little change in cytokine secretion as well. And the role of IL-6 has been kind of conflicting in the co-infection literature. In very severe co-infections, you do see an increase in patient bronchoalveolar lavage with IL-6, but its role is a little conflicting in co-infection. And so with these subtle changes, we became interested in what is the actual cargo of these vesicles, and maybe that could give us some insight into why they're not able to functionally clear Staph aureus in the setting of the antiviral program. And so to do these experiments, we decided to use a proteomics approach to understand what the cargo was. And so we collected extracellular vesicles from both control and antiviral treated epithelial cells and performed, in collaboration with Bill Haas's lab at the University of Pittsburgh, label-free quantitative mass spectrometry. And when we did that, I'm just showing you here the differentially expressed genes. We saw an increased abundance of a number of proteins, increased abundance of a number of proteins in the extracellular vesicles, but surprisingly no significant downregulation of any proteins. When we looked at the KEGG categories for these upregulated proteins in extracellular vesicles, these are labeled here at the bottom. But I just want to draw your attention to the blue proteins, and these were enriched in metabolic proteins. And so this really caught our interest because there's a growing body of literature in the macrophage field showing the metabolic state of macrophages is really important in dictating their antibacterial or antimicrobicidal versus a more suppressive or pro-resolving phenotype. And so this made us start to think about what the metabolic state might be of these macrophages, and maybe that's responsible for this functional change that we're seeing. And so as a quick review, when glucose is brought into the cell, it is moved through glycolysis to produce pyruvate. That pyruvate can then be shuttled to the mitochondria and used as part of TCA cycle for oxidative phosphorylation. It also could be converted to lactate as part of glycolysis. A glycolytic program in macrophages tends to be a more microbicidal or pro-inflammatory macrophage versus increases in oxidative phosphorylation are more associated with a suppressive or anti-inflammatory macrophage phenotype. So we went back and took a look at our proteomics. We saw that one of the most upregulated proteins in our extracellular vesicles was pyruvate kinase. And this drew our attention because pyruvate kinase is the last rate-limiting enzyme in glycolysis to make pyruvate that can then get shuttled to either oxidative phosphorylation or aerobic glycolysis. And so what we know in cells that there are 3 different forms pyruvate kinase can be in. PKM1 is an isoform. It's a constitutively active tetramer. It has high enzymatic activity in converting PEP to pyruvate. PKM2 isoform is when it's active, it's in a tetramer form, but it can be phosphorylated. Then it goes into a dimer form where it tends to be proglycolytic and an inactive state. So we wanted to understand what form of PKM2 was actually occurring in the extracellular vesicles and then how that was impacting the PKM function in the macrophages, because that might have a role in the ability to regulate metabolic state. Apologize for the quality here. If we look at the extracellular vesicle content of PKM2, so that's what we saw changed in our mass spectrometry work, we see the extracellular vesicles are only increased in PKM2, and this is only increased with the poly-IC or the antiviral extracellular vesicles. So this is kind of confirming what we saw by mass spectrometry. Now, if we move into the macrophages and take a look at the PKM isoform in macrophages, we see no change in PKM1 isoform. PKM2 is increased. Phosphorylated form of PKM2 is actually downregulated in macrophages with the antiviral extracellular vesicles. And so this is consistent with a reduction in glycolytic capacity and an increase in the oxidative phosphorylation or more suppressive state of macrophages. So we then wanted to ask, can we, so this phenotype is with antiviral extracellular vesicles. We wanted to really narrow in, is this phenotype of an increase in PKM2, so one of the single molecules are playing a really important role in and of itself in changing the metabolic state of the macrophages. So we used a small molecule, TEP46, which will increase PKM2 active state in macrophages, and wanted to ask, which we confirmed with in our own macrophage population, does this impair macrophage antibacterial activity? And so we performed an antibiotic protection assay here, and we show that TEP46, so just activating PKM2, is enough to impair the clearance by macrophages of Staph aureus, similar to what we saw with our antiviral extracellular vesicles. And so this suggested to us that what we're seeing in the macrophages that have been exposed to the antiviral extracellular vesicles is an increase in this active tetramer form of PKM2, which will result in an increase in oxidative phosphorylation and reduced microbicidal activity. So the last question then was, do we actually see a difference in the metabolic state of the cells? All the data would suggest we would see that. And so we used seahorse mitochondrial stress assays here and looked at oxygen consumption rate as a readout for oxidative phosphorylation. We see here in blue using TEP46 just inducing, and I'm quantifying here, in blue just inducing PKM2 to its active state can increase oxidative consumption rate and oxygen phosphorylation or oxidative phosphorylation. Similar, just applying the antiviral extracellular vesicles also converts or induces more oxidative phosphorylation in the macrophages. And this is largely due to an increase in the spare respiratory capacity as the underlying mechanism for this change in the metabolic state. So what I've shown you today is during antiviral signaling in the respiratory epithelium, they can release antiviral extracellular vesicles. These vesicles, when they interact with macrophages, can induce the cytokine production of IL-6. They can also increase the active state of PKM2, which will result in an increase in oxidative phosphorylation in these cells, and a more suppressive state, which will result in reduced bacterial clearance of bacteria like Staph aureus. When we think about this in the context of also the vesicles interacting with bacteria like Pseudomonas and improving Pseudomonas biofilm growth, this leads us to the model that this antiviral signaling by the respiratory epithelium during co-infection can impact multiple different pathogens and different mechanisms to promote the persistence of pathogens during co-infection, which is what we see in the clinic. And so this might be one mechanism by which co-infection can promote chronic infection. We're interested in focusing on metabolites that are being released and potentially associated with these vesicles that would mediate both macrophage function as well as Pseudomonas biofilm. And we're really excited by new developments in the cancer literature that show that there are a number of drugs are being developed to target glycolysis and various metabolic changes in cells that we could use in the respiratory epithelium to maybe change the profile of these vesicles being released as maybe a novel therapeutic strategy. With that, I want to thank everyone that did the work in the lab. This was, like I said, largely done by former graduate student Sydney Lane in the lab. Acknowledge my funding, and I can take any questions. Thanks. We think some of this might be a timing feature. It's probably you do want to promote resolution of inflammation eventually. And so we think it's probably a timing feature. There are also evidence, we haven't dug into the extracellular vesicles as much ourselves for this, but there are interferon stimulated genes and things that can be transported in the vesicles that would be carrying out an antiviral program to potentially allow the cells to clear the virus, but then also start the process of resolving inflammation. And that would be my best speculation of what's going on. Mm-hmm.
Any other questions in the audience?
Oh, and I want to remind people also you can submit questions on the app as well.
Yeah, if you want. I think there is a question in the back.
Can you yell a little bit louder? Yeah, so we've done a number, in the interest of time, I didn't show all of this. We characterized them using nanoparticle tracking analysis. We also did a bunch of different cell surface markers, and then some of the various sizes. So we did a number of different characterization methods that were required by the. So we've floated these vesicles on a gradient as well to try to purify them away from some of that extra debris, and that we see the same phenotypes with with doing that. And so that hasn't changed our phenotypes. But so part of the reason we use the ultracentrifugation, because I know there are other methods to use, is we found in the past using ExoQuick and some of these other methods to purify extracellular vesicles. The previous studies, we were looking at cargo that was carried on the outside of the vesicles, and those purification methods changed the pH, which then changed the cargo that we were trying to study. And so we've done a number of different methods to try to convince ourselves that these were in fact extracellular vesicles. We have EM also. The papers have all kinds of characterization in them that I just didn't have time to show today. So this was non-CF. These were non-CF MDMs. We haven't done the CF MDMs yet. We're in the process at Dartmouth of getting that set up. And so we would like to do that. I think especially understanding the metabolic state might be different in CF. And so I think it's a great question and we need to consider it. We haven't done it yet. Yeah, it's a great question. We've thought about that. We've looked at their interaction. They can interact with other epithelial cells. I think it would be interesting. Pushing oxidative phosphorylation may be a good way of actually controlling some of the viral infections. So I think that's a potential mechanism that could be interesting. I think in the context of our particular experiments, we were trying to isolate just the interaction with macrophages. But I do think in the actual airway, I think they could be interacting with a number of different cells. And I had something else to say, and I just lost it, so I'll tell you later.
There's another question here.
That's an interesting idea. I mean, they're not full from they may be full at a metabolic capacity, and that's something I think we need to do metabolomics and dig a little bit deeper here, and I think that's an interesting idea. They're not full from a space in the stomach of the macrophage capacity. We've done imaging, which I just didn't have time to show today, and there's definitely capacity to phagocytose more things, but I do like the idea of digging deeper in the metabolic state, as I said. I think we need to look a little bit deeper moving forward. So I like that idea. And the first sales talk was great and made me think of some ideas too, Tony. So
we have time for one more question, if there is one in the audience.
Let me think back. It was 6 mils of media. So we would use, 6 24 millimeter transwells per experiment is what we were doing.
Okay, well, thanks very much, Jennifer. Awesome talk.
Thank you, Jennifer. Now I'm going to present Dr. Sebastián Riquelme. He's an assistant professor of immunology at Columbia University, and he works with Pseudomonas infection, inflammation, and metabolism.
Okay. Well, thanks very much for being here. So today we will continue talking about tolerance. I think some of you are discovering this new word, tolerance to infections. So in this session, we're trying to learn a little bit more how metabolism is involved in this. And today I will be telling you a story. Well, I'm poor, I don't have disclosures. So we will be discussing a little bit more of how metabolism regulates this immune setting during inflammation in the lung. So as this is hardcore immunology, I will remind a little bit more about how this mechanism is activated, but already Jennifer and Sail, they did a very good job explaining how this works. So we all know that when an infection occurs in a mucosal tissue, the host responds to the infection through a mechanism that is called the resistance to infection, in where the goal of this resistance is to promote a massive and acute inflammatory response mediated through different pathways. And one of the most common one is the inflammasome, which is mediated by this metabolite, succinate, derived from the mitochondria, this transcription factor, HIF-1 alpha, and this pro-inflammatory cytokine, IL-1 beta. And the aim of the goal of this inflammatory setting is to promote and clear the infection. So just a little bit more of background about this pathway. Normally in macrophages and other myeloid cells, the inflammasome gets activated when the macrophage sends some PAMs, such as LPS, from the surface of gram-negative pathogens through surface receptors, such as TLR4. And this interaction promotes a signaling cascade that lands on the mitochondria. And the mitochondria reprograms its metabolism, accumulating different metabolites from the TC cycle. One of those metabolites is succinate, which is a minor pro-inflammatory metabolite. This succinate, by inhibiting proline hydroxylase, which is a negative inhibitor of this transcription factor, now releases the HIF-1 alpha factor, and HIF-1 alpha can migrate to the nucleus, where it promotes the transcription of the pro-IL-1 beta, the precursor of these pro-inflammatory cytokines. Then in the cytoplasm, certain caspase corresponding forming part of the inflammasome can promote the cleavage and the release of the bioactive form of this pro-inflammatory cytokine, IL-1 beta. And this IL-1 beta can activate the macrophage itself to kill the pathogen, or at the same time promote the activation of different myeloid cells around to promote the killing of the organism. So, however, sometimes we get infection with pathogens that are very hard to eradicate, such as, for example, the pathogens from the escape family, such as Pseudomonas aeruginosa or Staphylococcus aureus, or many other pathogens. And what happens here is that the host keeps fighting and fighting infection for a long time. And if this inflammatory response is not adequately regulated, we can develop an immunopathology, and at the end of the day, ends up damaging the airway or the lung instead of protecting us from this infection. So at this point is where the host activates disease tolerance. And the goal of disease tolerance is to suppress this detrimental inflammation that ends up promoting the immunopathology. And one of the things that we have found in my laboratory is that disease tolerance, what it does is that reduce this excessive inflammation by reducing the inflammatory properties of the pathogen itself. For example, reduce the expression of LPS in the pathogen, reduce the expression of different toxins. But the question that we have now, well, and this at the end of the day, when the pathogens they become less inflammatory, now the host stop attacking the pathogen, and the pathogen stop attacking the host, and this come into this agreement in where we call the host pathogen coexistent, which clinically is just an indolent infection or a long-term infection. So the question that we will try to address today is how this process occurs, how the host come into this agreement with the pathogen to not attack each other and to come into this coexistent along the time. So we will be focusing on the inflammasome, and particularly we will be focusing on another metabolic pathway that also is produced in the mitochondria of the same macrophages that at the beginning were fighting the infection. These macrophages, they can be pro-inflammatory, but then they can switch the pro-inflammatory phenotype and they can acquire a tolerogenic phenotype, as was described before. And one of those metabolites, it is metabolite called itaconate, which is synthesized by this enzyme aeruginosa. Some people say that itaconate is bactericidal, which we think that is not always the case, but what is very important for itaconate is that can suppress the inflammatory signaling promoted by succinate. So it's a mitochondrial metabolite that can regulate pro-inflammatory pathways mediated by the mitochondria as well. So said that, as a summary of this first part of this introduction, we know that the succinate HIF1 alpha L1 beta axis promotes resistance to infection, and that can promote tissue damage. But at the same time, itaconate later on during the infection, try to regulate that resistance to infection to decrease the immunopathology induced. And some pathogens, they can exploit this environment to persist and to coexist with the host. And this is the moment when we have to introduce Pseudomonas aeruginosa, because we know that Pseudomonas aeruginosa can last for years in the airway of people with cystic fibrosis, not attacking the host, and the host also does not attack the pathogen. And they are coming into disagreement. But we still don't understand how this process occurs. So as an introduction, we know that Pseudomonas CF is a genetic fatal disease caused by mutations in CFTR. People with CF exhibit increased airway inflammation, so they have an exacerbated resistant response to infection. And at the same time, the people with CF, they develop long-term lung infection with Pseudomonas aeruginosa. So the concept of disease tolerance comes to our mind. And one of the major characteristics of this infection by Pseudomonas, this long-term infection, is that this pathogen, they try producing biofilms, these communities. And the question is how? Are these communities associated with the activation of disease tolerance? And maybe the pathogen are exploiting the activation of this different type of immune response to the infection to survive and to thrive in the airway producing these communities. So the hypothesis that we will be testing today is that Pseudomonas persists in the CF lung through disease tolerance, through this alternative mechanism of defense to reduce immunopathology. So, as a first couple of results, we wanted to compare what is the type of metabolic response that Pseudomonas aeruginosa induces in the host during pneumonia, in the airway. So for that purpose, what we did is that we use a highly pathogenic bacteria that is a PO1, which is a classical laboratory strain of Pseudomonas aeruginosa, which everyone uses, okay, to mimic infection. And we compare the metabolic response against infection with a collection of 17 isolate of Pseudomonas derived from sputum of people with cystic fibrosis. Okay, these strains, they are already adapted to the airway, so they are already coexisting with the host and they are adapted to disease tolerance. And in comparison with PO1, we found that the Pseudomonas isolate, they induce much less accumulation of succinate in the airway, meaning that they don't trigger a metabolic response associated with inflammation. Instead, they promote much more itaconate accumulation. So they trigger the anti-inflammatory response, the anti-inflammatory program of the mitochondria through metabolism. And in fact, if we analyze the airway of people with cystic fibrosis, we can find that this itaconate also is enriched in sputum samples, meaning that this metabolite probably has a major role in the regulation of inflammation and at the same time in the production of these biofilms that coexist with the host. In fact, if we compare how much the infection by Pseudomonas depends on the production of itaconate by the host, we see that if we infect animals that can or cannot produce itaconate, the infection with PO1 is not dependent on the production of itaconate. However, if we infect with the CF isolates, we see that the infection by the CF isolate is highly dependent on the ability of the host to produce itaconate, meaning that these bacteria, they evolve in the airway and they become dependent on the presence of itaconate in the airway to survive. In fact, if we take one representative isolate from here and we interrogate them, can you produce biofilms with itaconate in comparison with PO1? We see that they produce at least twofold of more biofilms in comparison with our laboratory strain. So this Pseudomonas aeruginosa, it looks like they evolve in the airway to exploit this metabolite, this immunometabolite produced by the mitochondria in the host. As Pseudomonas is evolving and exploiting this metabolite, maybe Pseudomonas has a mechanism that can use to exploit and to induce much more production of this metabolite by the mitochondria. And the first question that we had is how Pseudomonas induces this host itaconate synthesis. And to answer that question, first we have to understand how the biofilms are formed, because maybe there is a component in the biofilms that is helping the bacteria to promote this mitochondrial response during the infection. And what we noticed that when the biofilms are formed, the biofilms are coated by different extracellular polysaccharides. And one of the most important extracellular polysaccharides in Pseudomonas is alginate. In fact, Pseudomonas coats a specific cluster that is called ALCH-D that promotes the synthesis of this extracellular polysaccharide, alginate. So the hypothesis that we had is that maybe this extracellular polysaccharide that is highly produced and secreted in these biofilms, maybe this extracellular polysaccharide is promoting or is reprogramming the mitochondria in the host to release more itaconate for their own benefits. So to answer that question, what we did is that we took animals and we infected with a wild-type bacteria or a bacteria that cannot produce alginate. And then we evaluated the concentration of itaconate in the airway and the production of inflammatory cytokines, mainly the ones associated with inflammasome. And linking with our hypothesis, we found that the infection produced by pathogens that produce alginate correlated with an increased concentration of itaconate in the airway, meaning that the production of this extracellular polysaccharide by the pathogen reprograms the mitochondria in the host to release this itaconate. And at the same time, this itaconate-rich environment suppresses the accumulation or the release of IL-1 beta, which is the cytokine that is associated with inflammasome, but not other pro-inflammatory cytokines from other pathways. And just as a control, so you can believe me that this is really controlled by itaconate, we did the same experiment, but with animals that can or cannot produce itaconate, and we found exactly the same, that only IL-1 beta is being suppressed during the infection, but not other cytokines. And all of this is telling us, so there is an axis in where the bacteria, after producing these biofilms through alginate, can exploit the ability of the host mitochondria to produce this immunometabolite to induce this tolerogenic response to the infection. But during this tolerance is something that we have learned in nature is that as the host and the pathogen are coexisting, so they establish some bidirectional interactions. And not only the pathogen will be promoting the host to produce itaconate, but also itaconate can end up telling the bacteria to produce more alginate. So we wanted to evaluate that second part of this cross-communication, this agreement between host and pathogen. So to test that hypothesis that itaconate is promoting alginate production, what we did is that, I have very beautiful colors here, I promise you, but this is this is the way how Pseudomonas produces alginate, okay? So just as a little bit of introduction, we wanted to know if itaconate is able to promote all these metabolic pathways that end up in the production of alginate at the end of the day. So normally, Pseudomonas to produce alginate, Pseudomonas activates this anabolic process that is called gluconeogenesis, in where from pyruvate and acetyl-CoA, the bacteria produce fructose 6-phosphate. this fructose 6-phosphate can be shunted into the production of really complex sugars or enters the pentose phosphate pathway. And then the pentose phosphate pathways interact with orotate, which is the rate-limiting step of this, the de novo pathway of pyrimidine synthesis, and produce different nucleotides, such as UTP. Now, this nucleotide, they can interact with these sugars and they can promote this nucleotide activated carbohydrate that can be taken or utilized by different genes that promotes these extracellular polysaccharides, such as ALG, to promote alginate at the production of biofilms. And the hypothesis that we had is that maybe itaconate is exploring or is inducing all these metabolic changes to promote alginate production. And we had that hypothesis because we know that Pseudomonas has a trick in comparison with other pathogens. Pseudomonas express a cluster of genes that can break down itaconate and allow the incorporation of itaconate into pyruvate and acetyl-CoA. So we thought that that was the entry for itaconate from the mitochondria to enters into the bacterial metabolism and take control of the central metabolism of the pathogen to force them to do what they want to do, what the metabolite wants them to do. And so you can believe me here, what it did, this is again another experiment that if we remove one of these genes, now the bacteria loses its ability to produce biofilms in the presence of itaconate. So this process is mediated by this cluster. So how can we demonstrate that itaconate is really taking control of the bacterial central metabolism? So what we did is that we incubated Pseudomonas with itaconate, with a carbon label itaconate. So we can track how these different carbon atoms from itaconate can enter into every single molecule in the central metabolism in Pseudomonas aeruginosa. And we track this through isotope carbon labeling. So the first thing that I will show you is how itaconate affects the abundance of the different metabolites from all these pathways. So many pathways, many names. So just to do a long story short, we see that when we expose Pseudomonas to itaconate, many metabolites involved in gluconeogenesis, they get enriched. Okay, so the bacteria responds to itaconate by increasing fructose 6-phosphate, glucose 6-phosphate, and glucose. And fructose 6-phosphate is super important because it's the one that triggers everything else. Then if we analyze, for example, these modified carbohydrates that are here, these sugars, also we see a major induction of these different metabolites by itaconate. Also, this mitochondrial metabolite induce accumulation of different intermediates of the pentose phosphate pathway, as well induce, for example, orotate, which I told you that it was super important for the de novo pathway of pyrimidine synthesis to produce these nucleotides. It's highly enriched. And also we observed that Pseudomonas responded to itaconate by accumulating different other nucleotide activated carbohydrates, which are very important for the production of extracellular polysaccharide. So these mitochondrial metabolites really is promoting in the bacteria the upregulation of all these metabolic pathways involved in biofilms production. But now the question is, are these molecules or is itaconate integrating into this molecule, yes or not? And for that, we analyzed the different isotopologues, okay, of these molecules, and we found that at least 70% of all the molecules of acetyl-CoA contain carbon atoms that derive from itaconate. So meaning that itaconate is really integrated into the metabolism of the bacteria. Something similar happens with pyruvate. Around 60% of the molecules from pyruvate, 60% of the carbon atom from these molecules of pyruvate, they contain carbon atoms derived from itaconate. The same happens with phosphoenol pyruvate, another major regulator of gluconeogenesis. G3P, fructose 1.6-phosphate, fructose 6-phosphate, and glucose 6-phosphate. You can see here that now around 90% of the molecules contain carbon atoms derived from itaconate. However, this is not a general process. It looks like it's a highly regulated process because when we analyze the incorporation of itaconate into glucose, nothing happens. So the bacteria allows itaconate to take control of its central metabolism, but not everywhere, only in specific pathways related to the production of biofilms. Then if we analyze other metabolites, for example, the ribose phosphate from the pentose phosphate pathway, around 80% of the molecules contain carbon atoms from itaconate, the same on orotate, uridine, on uracil, and so going on and on. We have more data, but I'm not showing everything here. So as a matter of conclusion, okay, it looks like during an infection with Pseudomonas aeruginosa, what is happening is that the host responds to the infection through an acute inflammatory response mediated by the inflammasome. But after time, the same macrophage or the same immune cell can polarize its immune response towards a tolerogenic phenotype, and where the mitochondria now start producing itaconate to try to suppress the excessive inflammation to mitigate the immunopathology associated with this immune response. And this itaconate that is being produced that is suppressing inflammation can integrate into the bacterial metabolism to promote the synthesis of these biofilms. And now this pathogen exploited this disease tolerance mechanism, and by producing this alginate, induce even more itaconic production. And now there is the establishment of this bidirectional interaction where you have a vicious cycle where itaconate promotes biofilms and biofilms promote more itaconate. And now the host and the pathogen, they come into disagreement in where the inflammation is suppressed and the bacteria also can produce these biofilms. So said that, and that comes into the immunosuppression, and the host and disease tolerance is activated, and there is the host pathogen coexistence that I was telling you at the beginning. So said that, I would like to thank my laboratory. Also, I would like to thank the collaborators at Columbia University, Dr. Alice Prince, also the funding, the Cystic Fibrosis Foundation, of course, and our collaborators. I'm happy to answer any question. I knew that this question was coming because, and I will say this, I haven't seen like strong evidence saying that alginate is a virulence factor. Alginate is used as a food preservant. Alginate is used as to promote in bioengineering to produce tissues and to promote tissue repair as well. And it's used in ointments and all of that. But you're asking about the mucoidy. So, So we repeat this experiment with the MuK22P01 strain, which contained the major mutation in cystic fibrosis that promotes this mucoid phenotype. And we see that when Pseudomonas aeruginosa has this MuK mutation, the pathogen infects more the airway, the lung, and the bowel. So we are thinking that maybe it's more pathogenic, no? Because more CFUs, the host should die. However, if we remove the operon, or we remove the gene that produces alginate, now the bacteria becomes more pathogenic. It's the opposite. So this fits with what we are saying. The production of alginate by the pathogen is good for the host, and that's why the host ends up exploiting this pathway. Now, let's analyze the cytokines. If we remove alginate from the bacteria, now we have way more accumulation of IL-1 beta and other pro-inflammatory cytokines, such as IL-6, okay? But not TNF-alpha. But what happens with the host? The host, if you infect it with wild-type P1, the host rapidly dies. But hosts infected with the MuK22, they don't die. They are very happy. And then if you do the infection without the alginate, the host die. So from where, under our perspective, in one moment, we assumed that alginate was something negative. Someone said that, and we just believe it. It was a clinical correlation. But we think that we have data that is saying the opposite. I have data with FRD1. So we know, you know that FRD1 is a mucoid strain that in cystic fibrosis produces a lot of alginate. So we did the infection, as you can see, just don't think just CPO1 because it's not the best control because they have way more mutations. FRD1 has like 9,000 mutations in comparison with PO1. But you can see that if we remove LGD from FRD1, now the bacteria is more pathogenic. It's exactly the same. And if you analyze the inflammatory cytokines, IL-1 beta is way more, and IL-6 and TNF-alpha. So I think that this is a strong evidence saying that the production of alginate is, at the end of the day, maybe it will be good for the host. And maybe that's the reason why we see all these strains that produce a lot of alginate in their way. And that makes sense with the idea that alginate is a good thing, and we are using it in bioengineering, in preserving food, and all of that. And it's FDA approved. So why we start thinking that alginate was a virulence factor? Yeah, no, we haven't. It's a very good question, but we haven't measured that. But we think that the modulators will have a high impact in the tolerance phenotype because they are able to modulate the inflammatory response. Okay, so we think that we'll have a higher impact. We haven't done any experiment related to that with this story, but we think that maybe there is a link. Yes, in fact, that is a very good point because we all have been like admiring this beautiful work done by Pradeep Singh, where they show that the bacterial burden goes 2 logs down, but at the end of the day, the patients are still infected. But the patients are fine; they are not sick. So if they're infected and they are not sick, maybe those bacteria that are still there are bacteria that are already established tolerance with the host. So do we care about killing those pathogens? If the host already figured it out through disease tolerance? Well, in terms of the in vitro experiment that we did, we just did it with itaconate like that. You can buy it, so in that sense, doesn't matter from where it's coming. But the major cell population that produce itaconate in the lung are the myeloid cells, and we have alveolar macrophages. But if you analyze through single-cell RNA-seq, there is a major neutrophil populations that produce pockets of itaconate, a lot of itaconate. Alice Prince already showed with the staph that these neutrophils, they play a major role in the production of itaconate and the staph regulation, infection regulation for staph. And we think that with Pseudomonas may be kind of the same. Yes, that's a very good question. And there are like 3 questions that I could read. First of all, itaconate can enter Pseudomonas because there was a paper just published a couple of months ago where they discovered the transporters. So there is a transporter for itaconate in Pseudomonas, okay, first of all. Second one, the electrophilic properties of itaconate, they are known. They can react, this metabolite can react with different cysteines in the bacteria, can modify different proteins as they do in macrophages, okay? And as part of this paper that we already have submitted, we have, I didn't show this data here, but we have strong data suggesting that itaconate, when enters the bacteria, before going into gluconeogenesis, produce post-translational modification of different proteins in Pseudomonas. And that changed the activity of those proteins and the fate of Pseudomonas biofilm self-emerge.
Okay, we have time for one more question.
Yeah, in fact, we did some experiment in the past in where we exposed Pseudomonas to itaconate, and we saw that itaconate suppressed everything that has to do with, for example, utilization of glucose and everything. So it looks like Pseudomonas stop using carbohydrates from the environment to produce these extracellular polysaccharides, and instead start producing everything through this new carboxylate, itaconate. Because at the same time, what I show is that people have said that itaconate is bactericidal, but the bacteria faces that. It's like a decision where do I use itaconate or I die? So it looks like the bacteria is forced to internalize it, to break it down into pyruvate and acetyl-CoA. If not, the bacteria will die through all this post-translational modification and all this stress. So it's like a selective process that at the end of the day converge into disease tolerance.
Okay.
I think, yeah, we're good?
Yeah.
Oh, okay.
Yeah, we're good.
Well, thanks very much.
Thank you.
So let me introduce you. So next is Dr. Neha Garg. She's an associate professor at the Georgia Institute of Technology, and in her laboratory, she works with host pathogen and host microbiome molecular interaction. And today she will be showing us her new advances.
Thank you for that kind introduction. Thank you, Hazel and Sebastian, for having me here. I've been really excited in this meeting. This is my first NACFC. I've been taking notes like a maniac. I've learned so much. I've been very excited to hear all the talks. I've learned a lot, and I'm looking forward to the rest of the meeting. All right. So my lab, I'm a chemist by training. So my lab applies chemistry-first approaches to understand microbe-microbe interactions, microbe-host interactions, microbe-drug interactions. Today, I want to give you a little flavor of how metabolomics is a foundational approach that allows us to understand biology, allows us to understand how host microbe interact, how they interact with the drugs, how microbe interacts. Today, in interest of time, I'll focus on microbe-drug interactions, but my lab also studies microbe-host and microbe-microbe interactions. So in my presentation today, I'll focus on Burkholderia cepacia complex that causes the least number of infections in CF patients. But it is considered the most feared pathogen in CF patients because it leads to cepacia syndrome, multi-organ failure. It also leads to epidemic outbreak between patients. In the past, it also led to patient and care team isolation because Burkholderia could spread between patients, leading to these epidemic outbreaks, sometimes also removal from the organ transplant waitlist. One of the challenge with Burkholderia also is that it leads to diverse infection outcomes. Now, when Pseudomonas causes an infection, it's almost always aeruginosa. When staph causes an infection, it's always aureus. But when Burkholderia causes an infection, 20 different species of Burkholderia can cause infection. There can be different species and different strains of those species that can lead to diverse infection outcomes. So we wanted to explore, can we identify a metabolomic signature that we could link to these diverse infection outcomes? To ask these questions, we study these microorganisms first in synthetic sputum media, which mimics the physical and chemical environment of the lungs. And we also expose these bacteria during culturing to antibiotics the patients might be taking. And then we look at the metabolomic and transcriptomics response. Today, I'll only talk about the metabolomics response. So for today's talk, I'll talk about different pathogens that cause infection. So these are the 3 species we studied, and we cultured at least three strains of each species. And all of these strains came from CF patients. And we exposed them to trimethoprim with no trimethoprim controls. And then we extract them and look at their metabolomics data. So our isolates come from CF patients. We culture them. We run them through a chromatography unit, which separates the molecules, and then we acquire mass spec data. Now, analysis of mass spec data involves 3 steps. First is data processing. We identify area under the curve for every molecule that's detected. Then we apply some multivariate statistics to identify what molecules correlate with the phenotype that we are studying. Then we rank them, and we try to identify them, and then we link them to the biological interpretation. So now this compound identification is a challenge in metabolomics because there is no code for metabolomics. For genomics, you have code as ATGC. For peptides, you have code as amide bond. But for metabolomics, due to their diversity of structures, size, there is no code that can be used for annotation of metabolomics. So how that's done in a mass spectrometer, you can isolate a molecule, break it into pieces. This is called fragmentation spectra, which serves as a fingerprint. You can look for this fragmentation spectra in databases. If you get a match, you can name your molecule. But using this methodology, you can name maybe 3% of the molecules you detect in your data. So think about just being able to annotate 3% of the genes in a transcriptomics data, right? So it's difficult. In my postdoctoral lab, we developed new methods to propagate these annotations, where if 2 molecules are structurally similar, their fragmentation spectra are similar. So we can quantify the similarity and represent the similarity in the form of these connected nodes. So these 2 molecules are structurally similar. They likely come from same pathway, or they are biochemical transformations. So using this strategy, if you can annotate this molecule, you can propagate annotations to additional molecules. I'll highlight some of these methods and in silico methods today in my talk to show you how metabolomics can enable discovery and understand interactions between different microorganisms. So first, we cultured, as I mentioned, 3 different species, 3 strains of each species in the presence of antibiotic trimethoprim. And then we look for species-specific cluster of molecules. They might be signature of infection outcomes, for example. So we identify one such cluster, which is only produced by B. Cynosephacia, and it was only produced in the presence of antibiotic. And we ran those fingerprints, the fragmentation spectra, to every single database that's out there, and we came back empty-handed, no annotations. We don't know what these molecules are. So then we applied another approach that was recently developed, where you can use language-based models to predict a common substructure. So you can't tell what the molecule is, but you may be able to tell what a substructure, subpart of that molecule is, or core structure of that molecule is. So using this approach, we identified that all of these molecules contain trimethoprim as substructure. That means but only Burkholderia Cynosephacia is modifying the antibiotic trimethoprim through a novel, unknown mechanism. So we thought we may be onto a path for discovery of new mechanism for antibiotic resistance, and we wanted to chase it down. So we asked, do all of our strains carry out these modifications? Turns out only 2 of them do, and the 3rd one does not. And we got lucky that the 2 that do modify trimethoprim were pigmented. They produce the brown pigment. And the one that does not modify the antibiotic does not produce pigment. So we looked into the pigmentation pathway, which starts with tyrosine. Normally, tyrosine is catabolized and enters a TCA cycle. But in pathogens that are pigmented, there is a mutation in HMGA such that the flux is altered, homogentasic acid accumulates, auto-oxidizes, and polymerizes to this pyomelanin pigment. So to test our hypothesis whether pyomelanin, this pigment, has anything to do with trimethoprim modification, we wanted to swap out HMGA between pigmented and non-pigmented strains and test antibiotic modifications. Turns out Joanna Goldberg had already done that. She had switched the HMGA gene between pigmented and non-pigmented strains. So we got the strains from Joanna, and then we exposed them to trimethoprim and looked for modification of trimethoprim. So turns out only pigmented, blue and orange, produce these molecules that are modified form of trimethoprim. Non-pigmented do not. So pigmentation has something to do with antibiotic metabolism. So then we thought, okay, so maybe pigmented bacteria have some components of central metabolism that are unique to them that are being added on to trimethoprim. To test that, we compared the metabolomes of pigmented bacteria with and non-pigmented bacteria and found that these molecules are only produced by pigmented bacteria. Again, we run their spectra through several databases, come back, and empty-handed, we cannot annotate any of these molecules. So, but we do notice that all of these molecules contain multiple sulfurs. So there is some sulfur chemistry, sulfur metabolism going on in pigmented bacteria that may play a role in this. So we looked for a common substructure across these molecules. All of them contain sulfur, and if you are a chemist and you look at the carbon to hydrogen ratio, you can tell they are aromatic compounds. So serendipity that the pigmentation pathway also contains aromatic compounds. So that led us to a hypothesis whether any of these compounds are involved in the modification of trimethoprim. To test that, we use isotope labels. So we used labeled tyrosine, which is labeled at 7 carbons. And to account for sulfurs, we used labeled methionine or cysteine. And then we look for incorporation of these labels into trimethoprim metabolism. So using this approach, we were able to identify a pathway for trimethoprim metabolism where tyrosine is converted to homogentic acid. I mentioned that this molecule accumulates in pigmented strains. And then methionine, we know, is converted to methane thiol. If you are the graduate student culturing these bacteria in the lab, you know it makes methane thiol because it smells really bad. Then this methane thiol is added to these 3 open positions on the aromatic ring. So you get this molecule called trimethylthiolated homogentic acid. This molecule was previously unknown. We discovered it for the first time. And this molecule undergoes decarboxylation addition to trimethoprim, leading to inactivation of trimethoprim. So chasing untargeted metabolomics data, species-specific signatures, we identified a new pathway for trimethoprim metabolism. And turns out pyomelanin production is common across pathogens. And in CF patients, often it is observed that Pseudomonas goes from non-pigmented version to pigmented version where it produces pyomelanin. So this may be a new function of pyomelanin that we've discovered. So you may know of antibiotic resistance mechanisms, degradation enzymes, beta-lactamase. To this audience, I don't need to introduce this, but we've discovered a new one where trimethoprim reacts with this molecule we discovered, leading to its inactivation. So now we have a labeled approach where we can identify this sulfur-producing chemistry produced by only pigmented strains. So we annotated a number of additional molecules that come from the same pathway. And these are antimicrobial, antifungal compounds. So by producing these compounds, pigmented Burkholderia can also likely control community structure in CF patients. So summarizing this part of the talk, I showed you an approach, metabolomics approach, to discover methylthiolated homogentic acid as a new metabolite that's not only involved in the inactivation of trimethoprim, but also results in the production of several bioactive compounds. So pigment production leads to unique metabolic profiles with enhanced biological functions. So this is where I give you a flavor of how metabolomics allowed us to discover new chemistry, new function that we could link to bacterial phenotype, that is color, pigment production, and validated these observations using genetically modified bacteria. So now I'm going to move on to second part of my talk, where I want to show you how antibiotics at sublethal concentrations act as signaling molecules. So antibiotics, we know at high concentrations can be static or cidal, but I'll show you at low concentrations, they act as signaling molecules. So this is again metabolomics data of 4 different strains of B. Cynocipacia, either exposed to trimethoprim or not. And then this is metabolite clustering. We see clusters of metabolite that are only produced in the presence of trimethoprim. And they vary across strains. So this cluster is only produced by this bacteria, this one is produced only by this, and this one is only produced by this. So this suggests that this is not central metabolism. This may be specialized metabolites like vancomycin that bacteria produces. So to annotate these molecules, again, we apply several methods. All these 4 molecules are only produced by K56.2 in presence of antibiotic. We couldn't annotate them. Then in 2020, a new tool came out called Canopus, which can classify the chemical class of a compound. It can tell you what a compound is. It can tell you what a core structure is. It can tell you it belongs to this class of compounds. So this tool predicted it belonged to pyrazine subclass of compounds. Then we do a specific search for pyrazine ring-containing compounds produced by gram-negative bacteria, and we come across these compounds. And this is the pathway that is present in K56.2 for the production of these compounds. So these compounds, in our case, are only produced in the presence of antibiotics. They are not produced in the absence of antibiotics. And these compounds have been described in animal pathogens such as Pseudomonas entomophila and Pseudomonas syringae. And if you knock out these pathway in these plants or animal pathogens, it reduces their virulence. And in case of CF patients, taking trimethoprim as an antibiotic, these molecules are produced in the presence of antibiotics. So antibiotic at sublethal concentration is increasing their virulence. Another example here is production of this compound called fragin. It is a siderophore, it binds copper, and it's an antifungal compound. Again, it's produced in the presence of antibiotic. It was described in 1967 as plant growth inhibitor. We don't know what it does to mammalian cells, but at least we know it produces an antifungal compound. So now how is trimethoprim influencing the production of these compounds? People who work with Pseudomonas and Burkholderia all know about quorum sensing. So quorum sensing is one way which bacteria controls gene expression. In Burkholderia, there are 3 such systems. So there is a small signal called C8AHL, which when bound to its receptor, controls gene expression, or C6AHL bound to receptor controls gene expression. Unique to B. cynocepacia, there is an orphan receptor called SEPR2, which does not have a cognate ligand to bind, but it still controls gene expression. So we wondered whether QS is involved in regulation of production of these compounds in presence of trimethoprim. So we knocked out each of these genes individually and asked this question. Shown here is a knockout for SEPR2, which is the orphan receptor. So wild-type bacteria does not make the molecule without antibiotic, makes it only in the presence of antibiotic. And if you knock out SEPR2, the orphan receptor, it does not make the molecule, whether you have antibiotic or not. And if we complement SEPR2, we still again see the same phenotype as wild-type. So some of this induction of specialized molecule production is mediated by this orphan receptor. I also mentioned that they have a C6AHL and a C8AHL mediated quorum sensing. So when we knocked those out, we found that C6AHL is not involved. So without trimethoprim or AHL, we don't see production in the knockout. If we add C6AHL, we see a little bit enhancement in production. But if we add trimethoprim, we see the production. And if we add both AHL and trimethoprim, we see production. So this system is not involved in the antibiotic induction. But if we knock out C8AHL synthase, without antibiotic or AHL, we don't see production. With antibiotic, we don't see production. So that means C8AHL is involved in this phenotype. And it's not just AHL, because if we add AHL, we don't see induction. We need both AHL and trimethoprim to cause this induction. So this suggests that there may be a secondary global transcriptional factor that may be involved in this phenotype. So we knocked out several transcription factors. Turns out SCMR is involved in this phenotype. So if we knock out SCMR, that induction in presence of trimethoprim goes away. If we knock out MFTR, it doesn't go away. So if we knock out Shiva, it goes away a little bit. So these transcriptional regulators are involved. So this suggests and leads us to propose a potential model for trimethoprim response where quorum sensing is involved. C8AHL likely binds to the orphan receptor, activating the production through SEPR2. And then also trimethoprim is known to enhance production of xanthine, which serves as ligand for global transcriptional factors. So one of these transcriptional factor bound to xanthine is also required for this induction mechanism. So now trimethoprim enhanced the production of antifungal compounds by Burkholderia. So that led us to a new path to start probing bacterial fungal interactions in presence of antibiotics. So we did that. This is Aspergillus fumigatus strain cultured here, Burkholderia here. They happily coexist without trimethoprim. But if you add trimethoprim, Burkholderia starts inhibiting. And as you increase trimethoprim, you see complete inhibition. And then if we knock out that HAM-C gene that is involved in the production of this antifungal compound, then we do not see inhibition in absence or presence of antibiotics. So this suggests sublethal antibiotic concentrations, which are present in CF lungs, influence how bacteria and fungi interact with each other. So we started looking at fungal bacterial interactions in CF lung. We did co-culture or monoculture, and we look at molecules that are only produced in co-culture. So these 155 molecules were only produced in co-culture in the presence of, or in the co-culture in the presence of trimethoprim. So I'm going to give you a couple of vignettes of this story where we see how this co-culture affects their production of virulence factors. We also use imaging mass spectrometry, where we can take a picture of a Petri plate with imaging mass spec and visualize where the molecule is on the Petri plate. So this is an imaging mass spec image where on top, no trimethoprim. This is monoculture of Aspergillus, and this is co-culture. So we don't see production of this molecule, which is a siderophore iron acquisition compound produced by Aspergillus. So we don't see any production in monoculture. In co-culture, we see a little bit production, and if you add trimethoprim, we see enhanced production. So we did this in liquid culture also. We see the same phenotype, no production in monoculture, a little bit production in co-culture, and enhanced in the presence of trimethoprim. So we wanted to ask, how does trimethoprim regulates interactions of bacteria and fungus with each other? I think I'm missing a slide. But we knocked out HAM-C, the gene which results in the production of fragin, the antifungal compound I showed. And then if we knock out that fragin pathway from Burkholderia, we do not see this phenotype. So we show that fragin is involved in induction of siderophore production by fungus. And serendipitously, we also landed on a new phenotype where trimethoprim does not inhibit Aspergillus, but it inhibits the blue color of the conidia of Aspergillus. So without antibiotic, we see blue color, and with antibiotic, we do not see blue color. So this is the pathway involved in the production of this blue color. It's a DHN melanin, which is an important virulence factor. It has been explored as an antifungal target because it's required for pathogenesis by the fungus. So then we acquired knockouts in each of these genes from Dr. Chung, and in absence of trimethoprim, you see these beautiful color panel. But if you add trimethoprim, you see reduction of color. So this is again a serendipitous discovery. We are looking at the exposure of these bacteria or fungi to antimicrobial compounds. We discover how these compounds affect various different phenotypes. Now we are also looking at antifungal compounds to study how antifungal compounds affect bacterial fungal interactions. To summarize, today I showed you, and I really hope I gave you a flavor of how untargeted metabolomics allows us to link chemistry with phenotype, how that chemistry is regulated, and that allows us to peek into the function of those phenotypes. So really how metabolomics allows us to connect phenotype, chemotypes, and genotypes, and allows us to do discovery, really is shaping our understanding of cell physiology, morphology, microbiology. With that, let me close my presentation by saying we traverse the same trajectory of omics, just in the opposite direction. We start with metabolomics, we discover new phenotypes, we generate hypotheses, refine hypotheses, validate hypotheses using other omics methods. With that, I have the privilege to present the work today, but let me thank the people who did the work and our collaborators, Marvin at Georgia Tech, Joanna from Emory, and our collaborators at Sandia National Lab. Thank you for funding and for your attention.
We have a question over there.
Robbie?
You.
It's an DHFR inhibitor.
So I would imagine that microbes have not encountered it. What are your thoughts?
Yeah, so melanin production is observed in the environment also. It offers protection from harsh UV light, heavy metals in the environment. It's basically a polymer that sequesters a lot of toxic stuff. So what I didn't have time to go into details today is that modified trimethoprim actually gets incorporated into the pyromelanin pigment. So bacteria takes trimethoprim, takes that molecule that is involved in pyromelanin production, links them, and throws trimethoprim out into melanin production. And this is limited by how much methionine and tyrosine you have. So this also explains why antibiotic susceptibility profiles are different in vitro and in vivo. So in cell culture media, you are limited by how much methionine and tyrosine you have. But in a mucus of CF patient, you have a lot of tyrosine and methionine, and bacteria can continuously churn out this reaction, inactivate trimethoprim, and have higher resistance. Now, we have tested a couple of Aspergillus strains that we received from Emory University that were isolated from CF patients. We see the same phenotype. Yeah, we had a project with Stephen Dolan. We did that with him for Pseudomonas, and we see a shift in metabolites, but I'm not too familiar with the data analysis as of now, but for this work, we haven't. We should, I agree.
Okay, I do have one question here. So something that caught my attention were all these sulfur-containing compounds that you were discussing. So in the host...
in the bacteria.
Yeah, no, but my question is, in the host there are many like antimicrobial molecules that go and they interact with cysteine molecules from organisms.
Right.
So do you think that these molecules are like acting as sinks that can be like competing against it, providing some evolutive advantage against the immune response?
Absolutely. These are very, they are also redox active. So they would play, chemists discover antibiotics by culturing bacteria. So we associate function to molecules as something that can kill a bacteria or fungi. But in vivo, I think they play additional important roles. For example, fragin, that's an antifungal compound in vitro, but in plants, it has been shown to inhibit cell growth. Other siderophores in gut have been shown to also inhibit fibrosis. So we see enhancement of siderophore production by these pathogens in response to antibiotics. And so this is cystic fibrosis. There's a lot of fibrosis in there. And I wonder if these compounds also enhance fibrosis in the lungs as an in vivo function of these molecules.
That's a
Absolutely.
super interesting point. And the other question that I had is that, is it important what the bacteria, these organisms are eating to end up producing these compounds in the presence of these antibiotics?
Yes. So for example, the trimethoprim modification or all the sulfur-containing compounds, they come from cysteine and methionine. So we see the MIC values of trimethoprim is different in vitro and in vivo. Also in vitro, we've noticed if we add external methionine and tyrosine, they are more resistant to antibiotic because they can continuously churn convert trimethoprim to this inactivated trimethoprim. So absolutely, yeah.
Okay, well, thanks very much, Nikki.
Thank you.
Amazing.